Hello students, I am Ms. Diksha Gaonkar, Assistant Professor in Botany from Government College of Arts, Science and Commerce, Kepe. Coming to program, Bachelor of Science, 3rd year, Subject, Botany, Course Code BOC 106, Course Title, Cell Biology and Plant Biochemistry. Title of the unit, Biomolecules. Module name, Structure and Properties of Oligosaccharides, under that Sucrose and Maltose. Outline, Introduction to Oligosaccharides, Types of Oligosaccharides, Structure and Properties of Sucrose and Maltose. Learning outcomes, at the end of the module, you will be able to define carbohydrate, oligosaccharides, classify types of oligosaccharide, list the properties of sucrose and maltose, draws the structure of sucrose and maltose. Coming to the introduction of oligosaccharide. So, term oligosaccharide comes from the Greek word. So, here in Greek, oligo means few and saccharide means sugar. Oligosaccharide consists of short chain of monosaccharide units, that is from 2 to 10, which are joined by glycosidic bond or glycosidic linkages, which are formed when OH group of one sugar reacts with the OH group of anomeric carbon of another sugar unit. So here are the different types of oligosaccharides we have. So depending upon the monomeric units. So if there are 2 monomeric units present, it is disaccharide. If 3 monomeric units are present, it is trisaccharide. If 4 monomeric units are present, it is tetrasaccharide. If 5, it is penta, and this will go on. 6 hexa, 7 heptasaccharide, and this will go on. So to cite an example, under disaccharide, we have lactose, which is found in milk, sucrose in honey and seeds, maltose in seeds such as wheat. Then under trisaccharide, we have example raffinose found in beans and cabbage, raminose found in microalgae and diatoms, mannitariose we have found in the yeast. Under tetrasaccharide, we have stachyose in human milk and verbiscose in case of pentasaccharide, which is found in the legumes. So, these oligosaccharides are further subclassified based on the presence or the absence of free reducing groups, that is aldehyde or ketone. So these are classified into reducing sugars and non-reducing sugars. Now let us see what are reducing and non-reducing sugars. So we can say any sugar or any carbohydrate is reducing when it is having the free aldehyde or a ketone group. Okay, and they are in hemiacetal or hemiketal form. Example to cite under this is maltose. Coming to the non-reducing sugar, aldehyde or the ketone group is not present in the free state, but instead it is utilized in the bond formation. They are in acetal or ketal form. To cite an example, we have sucrose. Coming to the properties of sucrose. Chemical formula of sucrose is C12H22O11. So here we have 12 carbon atoms, 22 hydrogen and 11 oxygen atoms present in the chemical formula of sucrose. Molecular weight, it is 3.342.30 grams per mole. Density is 1.587 grams per centimeter cube. Physical appearance, white crystalline solids, and melting point, it decomposes at 459 Kelvin. Some more properties of sucrose. Sucrose is white. It is crystalline solid and it is soluble in water. So you can dissolve this sucrose in water. It exists in 2 crystalline forms. Now which are the 2 crystalline forms? Number 1, a stable form which is referred to as sucrose A, and its melting point is 184 to 185 degrees centigrade. The second form or second crystalline form of sucrose, we have the unstable form. It is referred to as sucrose B, and its melting point is 168 to 170 degrees centigrade. When heated, above its melting point, it forms a brown substance and it is referred to as caramel. Sucrose is sweeter than glucose. It forms sucrates with alkali and alkaline earth metals. Sucrose can undergo combustion reactions to yield carbon dioxide and water, that is CO2 and H2O, carbon dioxide and water respectively. Coming to the structure of sucrose. So, let us see here also how the sucrose is formed. Okay, so you can see here the glucose molecule as we have previously seen in the previous module. So, the structure of the glucose, and this is the structure of the fructose. So, when these both react with one another, the bond, the glycosidic bond which is formed between this glucose and fructose, we get the formation of the sucrose molecule. So, sucrose composed of glucose and fructose molecule, and in between them the bond that is present here, the glycosidic bond which connects them both. Now, let us see sucrose on hydrolysis. With the dilute acid or the enzyme invertase, it gives a mixture of glucose and fructose. So, here is the reaction. So, this is sucrose molecule. And when the reaction occurs, the hydrolysis occurs, that is in the presence of water. Okay. So, here we get a separate monomeric unit of glucose and fructose. So, this is after hydrolysis reaction, when the glycosidic bond which is present between the glucose and fructose is broken down, we get separate individual unit of glucose and fructose molecules. Coming to the second example under oligosaccharide, that is maltose. Let us see what are the properties of maltose. So, the name maltose, the other names which are referred, malt sugar or malt biose. Chemical formula using C6H22O11. So, 6 molecules of carbon, 22 molecules of hydrogen and 11 molecules of oxygen are present. Appearance, it is white, it is powdery, or sometimes it may exist in the crystalline form. Appearance, we have seen density is 1.54 grams per centimeter cube. Molar mass, we have 342.3 grams per mole, and solubility in the water, it is soluble in the water. So, maltose again, the white crystalline solid with the melting point of 160 to 165 degrees centigrade. It is soluble in water and it is dextrorotatory. Because of the presence of free aldehyde group, maltose acts as a reducing sugar as we have previously seen, and it is a dimer that is derived from the starch and glycogen polymers. And it is a major product of enzymatic hydrolysis again on starch. Its melting point is 160 to 165 degrees centigrade. Occurrence of maltose, where it is present. So, maltose does not occur abundantly in the nature. It is a major product of enzymatic hydrolysis on starch, and the sprouting seed grains are the rich source of maltose. Example, sprouting barley. Then sprouting cereal grains are rich in these enzyme amylase which can split the starch and dextrins, which can split the starch into dextrins and maltose. Coming to the structure, so here we have glucose and glucose units which are combining here, and it undergoes condensation reaction during which water is given out as a byproduct, and it results in the formation here. It results in the formation of something called as glycosidic linkage or glycosidic bond. And it results here finally in the formation of maltose molecule. So, maltose is made up of glucose and glucose with the glycosidic bond in between. So, in maltose, the glycosidic bond is between carbon number 1 of one glucose and carbon number 4 of another glucose molecule. The free aldehyde group can be produced at C1 of the glucose, so 1 of the glucose molecule in solution, and hence maltose is a reducing sugar. So, this is hydrolysis reaction. So, how the maltose when reacts with the water, when it undergoes this hydrolysis reaction, it gives or it gives us separate unit, that is glucose and glucose. So basically the bond that is present between the glucose and glucose molecule, the glycosidic bond, it gets broken out, and finally we get the formation of two separate monomeric unit, glucose and glucose. So that is been what explained here. So we get two molecules of alpha D glucose. Summary, we have learnt about oligosaccharide, properties of oligosaccharide, under that sucrose and maltose, structures of sucrose and maltose, and these are some of the references. Thank you, students.